So the first question is to Dr. Flume and to me. So I'll start with you, Dr. Flume. How do you know when a therapy is not working, and when do you take patients off of a product if you've decided it's not working?
Well, that's a loaded question. We'll see if we can walk through this. So with every medicine, as I told you, that these drugs come with their own baggage. And so when we're talking about treating these patients with a chronic therapy, the first issue we're trying to work towards is, can they tolerate it? Are there any problems? And with all of these medicines, you can't expect that 100% of people can take them. So obviously, if a person's having trouble with the medicine, we're stopping them. An easy example there is an aerosolized therapy can actually make people cough or have wheezing or chest tightness, and that's just, the complications can't outweigh the potential benefits. The other part, though, is how do you decide when a drug isn't working anymore? So whether you're putting someone on pulmozyme or inhaled antibiotics or chronic macrolides or these CFTR modulators, how do you know when they're not doing what you want them to do? And some of this is a little bit easier if I started, how do you decide when to start a drug? So here I'm talking about treating patients earlier and earlier, younger. So using therapies when you're not measuring lung function and so forth. And it's not an easy thing to sort of figure out. You know, how do you know if pulmozyme is working in a baby? Some are easier. For example, if we're going to use an inhaled antibiotic like tobramycin, well, they should have Pseudomonas in their cultures. That makes it easy. And we actually adopt a strategy of trying to eradicate that early infection before chronic infection is established. The standard teaching that infection, once it sets in, is always going to be there is being tested now, because even with the introduction of, say, ivacaftor, we're seeing the prevalence of some pathogens be decreased, which is of great interest. At my center, if we have patients who have grown Pseudomonas or on inhaled antibiotics, if they haven't grown Pseudomonas after a couple of years of being on drug, we actually are talking about taking them off of that, as long as we have some evidence that you know, they've had culture negativity. That either means that it's eradicated or else it's just been suppressed so low you can't detect it. So in those patients we've taken off, we monitor. Some we have to put back on because Pseudomonas grows again, and others we haven't grown it, so we continue. So I do think it's a conversation to have with patients about when to consider stopping these medications, and just making sure that if you do so, that you monitor to look for any adverse effects. I will tell you one that I do worry about, though. There were some interesting cases presented at the CF meeting last year, one of the fellows from UNC, of patients who are on ivacaftor for clinical indication. They had the indicated mutations, and the patients came off drug, not because of poor adherence, but because of pharmacy hurdles. And within a week after being off the drug, they had severe exacerbations where their lung function just tanked. And it got better once they got back on ivacaftor. And so for our perspective, it was an interesting conversation as to what the mechanism might be. But what it did is it made it very worrisome that if you have these effective therapies, that it makes it very difficult to want to take them off of them. Now, a more challenging piece, I think, is perhaps with the Orkambi experience, and you don't have that same robust response of lung function like you see with Kalydeco. That's always an easy sell when a patient has a marked improvement. What do you do when the lung function doesn't change? Does that mean the drug isn't working? That's where the subjective comes into great use, because sometimes the lung function doesn't change much, but the patient comes in and says, my stomach has never felt this good. And it's a hard thing to appreciate, to what it's like to go through a lifetime where you have cramping and bloating every day, all day long. And so when someone comes in and says, this has made a huge difference for me in terms of quality of life, it's hard to want to say, well, I'm sorry, it didn't change your lung function, I'm going to take it away. So we've tried to develop methods of monitoring that. And so my advice to people is, don't just stop drugs. That's the question patients always ask. If you say, I'm going to put you on Orkambi, they want to say, well, what can I stop? And the answer is, let's do one thing at a time. And if you choose to stop a drug, then just monitor to make sure that they're not having any other signs of deterioration before you, I wouldn't say see in 3 months or see in 6 months. And that's the key, is that this is really like a partnership. You've got to let the patients know that this is a conversation you have to have. Let's figure it out. Tell me what's working, tell me what's not working, and we'll figure out what the right combination is.
Thank you, Dr. Flume. And from the health plan perspective, it's really communication with a specialist to determine how is a patient responding. Sometimes it's simply just that reauthorization process, 3 months, 6 months, annually, whatever it might be. If there is an objective measure that can be applied, we could certainly ask for that. We could ask for the results of a test or something, but it certainly is challenging. We really rely on the patient-physician coordination, if you will, in that relationship to determine that. Our next question is for Mary. How often are guidelines updated? Is it per acquired data from the registry?
So it's a great question, and I would say with the breadth of things that are coming out in CF, perhaps not as frequently as we would like, because they are robust efforts that take time. So you would note, for example, Orphan is currently, the pulmonary guidelines are being updated to advise on Orphan, but it is not officially within the guidelines right now. So that's just an example. We certainly do take a look at updating. They're not by drug. They're by sort of notion of things like pulmonary guidelines. Mental health guidelines is something that was recently released. We just updated newborn and infant care guidelines. So when new drugs do come out, we try to bring the external experts together to take a look at the evidence and incorporate the new therapies as soon as possible, but it is a pretty rigorous method, and it does take some time before they are updated.
I can add to that, since I chaired the pulmonary committee for guidelines. We set up 4 sets of guidelines and actually wrote the SOPs for how we would move forward. And a key thing with all research is you have to disseminate. You got to get it out there, and guidelines don't help you unless you get people to do them. So the foundation was actually instrumental in trying to push those. We actually put them in our metrics, and so they're actually, the data we get out of the registry are often to address how often centers are following guidelines. But guidelines are intended to be living creatures. Mm-hmm. And so, as Mary said, it does take an awful lot of work. One of the first things I did when I started that was I had to plan my exit strategy because it is an enormous amount of work. But you also want to make sure that there is a sufficient amount of new evidence upon which to base guidelines. So there are some guidelines out there that might seem a little stale, like we have guidelines on airway clearance therapies, but I can assure you there hasn't been anything really novel that would really have an impact in changing those. So you don't want to spend a lot of time. It's not going to change practice at all.
Thank you. From the health plan perspective, we look to guidelines to try to create our own policies. So if there are not sufficient guidelines in the market, sometimes we may get it wrong, which then creates a challenge for you because we're sort of interpreting the data perhaps with a lack of the level of expertise that you have. So it's very helpful for us to use the guideline as a tool to create our own internal programs and guidelines as to how we might want to control access.
Can I just add one more thing to that? Because to us, the guidelines are the gold standard. That's what we'd really like to see. But as Patrick said, sometimes the evidence does take time to accumulate. And it's important that it be looked at robustly. And from it, again, we really want to strive to an evidence-based guideline every time we can. But one of the things the foundation does do is provide things like white papers, an amalgamation of some of the clinical studies around the trial data and adding what we can share about the experience before the guidelines are finalized. We have done that in a robust manner for both of the correctors, the modulators that have come out, as well as some of the other therapeutics, and tried to have an interim step that we can engage with payers about and be able to give them access to the clinical experts and sort of see how things are unfolding with some of these new therapies. And it's, I think, by and large, been a useful dialogue for all parties.
Thank you. All right, we're running out of time. I have one last question here. It's a pretty interesting one. With the number of gene therapy or genome editing technologies entering clinical development, especially in rare diseases, how can health plans best prepare for this new era of personalized medicine? What potential challenges do you anticipate? So the promise of gene therapy is really interesting and a bit scary because if you think about it, what would the cost of a gene therapy be, right? So if we have a treatment that might be $250,000 a year, and you have a gene therapy that in theory could correct the disease defect, what would it be? Would it be $1 million for the gene therapy? And if so, how does a plan absorb that cost? And I think the financing of gene therapies is going to be very interesting when we figure it out. Do you handle it as something like a sort of an annuity, where maybe whoever has the patient at the time, of the period of time you're going to say the gene therapy would be good for? So let's say it is $1 million, and you decide, well, we're going to kind of amortize it over 10 years. So whoever has that patient for a year pays $100,000 of that cost. That's okay, except if the patient moves to a plan that has a different approach to covering that gene therapy, you got a bit of a problem. Do we have a pool of money that everybody pays into? So maybe the government creates a pool, and everybody pays into that pool, and the pool pays for the gene therapy on an annual basis or something like that. It's really a challenging area. I don't know if, Patrick, if you have any ideas on how that might be approached, since you indicated there is a gene therapy in the pipeline here.
Well, the gene therapy is unfortunately is not close to being approved. The last study was done in the UK, and I don't know that there will be another one, but there might. DNA editing, which would be curative, is got some hope. The other therapies are not curative, so if you're doing editing of messenger RNA or using modulators, amplifiers, these would be a chronic, persistent therapy. So, as you know, the cost of a drug is often not a surprise, because you know what your market is, and you know how long it's going to be used and what the patent life and so forth. And so one of the important parts is competition. So a problem that we're facing right now is, so we have 2 drugs out on the market, and they were successful, and as you know, they're very, very expensive. How do we develop the next better drugs is our goal. And that comes at considerable cost, because your pool of patients to study might have just gotten smaller, the complexity of studying those patients, or if you have to do head-to-head comparisons, the cost of purchasing the other drug makes it very unattractive for industry to do that. And so we actually have had meetings, the foundation organized with the FDA, in terms of how do you develop it so that you can continue to develop competition and develop what could be a better drug. Now, the thing we face is, as you saw in my disclosures, I do a lot of clinical trials, and one of the lessons I've learned over time is it doesn't matter how good a drug looks, it's still not a drug until you finish your studies. And so if all of these drugs hit, that's a good problem to have for us, right? Mm-hmm. Heaven forbid, we have lots of choices that we can work with patients to see what might work best. Some of them aren't going to make it, but the science is promising, and now we have to do the hard work, which is to find out what are the important aspects that, in terms of safety, in terms of efficacy, and then which of those drugs are going to finally make it to market. That makes it hard to predict, because that entire pipeline might make it, or half of it might make it, and then you've got the competition of how a messenger RNA editor competes with a modulator. Or what if I've got a corrector and I want to add an amplifier, because that takes me up another notch in terms of efficacy. Ideally, you try to do things that it makes it so that the other costs could be reduced in terms of hospitalization costs or other medications. And so treating these patients early might make a big difference in the long grand scheme of things.
Great. Well, thank you very much.